Welcome to The Mind, the Cure Protocol for July 19, 2026. We begin with what changed, not merely what was announced. One movement in this path has already reached care. The rest are research signals that could open the next path, but each still needs proof. We will start close to real people, then trace the evidence back to the targets and questions that matter next. This protocol is a living loop. People bring the questions that matter. Fresh evidence changes the map. The system remembers what we have already seen, so an old announcement does not keep pretending to be new. Every signal ends with one practical next action, the experiment, validation, collaboration, or care decision that could move it closer to real benefit. FDA-approved Lipfendra, enlicitide, a once-daily oral PCSK9 inhibitor, to reduce LDL-C in adults with hypercholesterol. Phase 3 trials reported placebo-adjusted LDL-C reductions of 56% and 59% at week 24. For people within the stated indication, this is now a regulated care option. Treatment decisions still belong with patients and clinicians. Approval establishes regulatory status for the stated indication, not proof of superiority or the right choice for every person. Next, what real-world uptake, safety, and outcomes within the approved indication? FDA granted traditional approval to Fabhalta, iptacopan, to slow kidney function decline in adults with primary IgA nephropathy. Thaplau's IgAN reported 48% slower eGFR decline versus placebo over 2 years. For people within the stated indication, this is now a regulated care option. Treatment decisions still belong with patients and clinicians. Approval establishes regulatory status for the stated indication, not proof of superiority or the right choice for every person. Next, what real-world uptake, safety, and outcomes within the approved indication? PLOS Pathogens reports that RS17053, a reproposed 1A adrenoceptor antagonist, bound class A penicillin-binding proteins and eliminated intracellular brucella in vitro. Mouse infection models showed lower organ bacterial counts and improved survival. If confirmed, this could give antibiotic teams a better starting point against resistant infections. The useful part is a more practical route to treatment. The chemistry still has to survive scale, safety, and real bacteria. Next, reproduce and scale the chemistry, then test the antibiotic against resistant bacteria. AlphaGem combined structural alignment, protein language models, and deep learning mining to build genome-scale metabolic models. It was validated across diverse organisms and automatically reconstructed models for 332 yeast species. If confirmed, this could help drug teams test better molecular ideas sooner instead of searching blindly. The useful part is a smarter starting point, not a finished medicine. Biology still gets the final vote. Next, show with independent benchmarks and laboratory work that the targets produce better drug candidates. The practical read is simple. These are signals, not promises. But each one is a concrete target that researchers, clinicians, engineers, funders, and the people most affected can connect to a next step. Keeping the evidence visible is how the Cure protocol stays current without pretending the work is finished.